of minutes for people to log on, but we're moving past 11. So we'll get started with this morning's Research Bites from UCD's Institute of Food and Health. So thank you all for joining us. And my particular thanks to Pilar Marcos, who is going to speak to us today and deliver the Research Bites coming from the pillar of food safety of the Institute of Food and Health. So I'm delighted to welcome you this morning. So Pilar Marcos, her degree is in Food Science and Technology with an MSc in Food Quality Safety and Technology. And all of these were attained at the University of Zaragoza in Spain. And indeed, the Institute has very long links with that university, providing us with very fine students and postdocs over the years. So her work is being done in Chagas Ashtown, where she's supervised by Declan Bolton. And on the UCD side, she's supervised by Professor Paul White. Her research is focused on Clostridium difficile and its foodborne transmission to humans. And I suppose the question that she is trying to get more knowledge in, or the gap that she's trying to fill, is about the prevalence of the bacteria in the food chain and also analysing its virulence. She's also studying how Clostridium difficile behaves in different foods that present different characteristics. And I suppose the key element of it is understanding if the presence of this microorganism in food is of a concern from a public health point of view. So over to you, Pilar, if you want to start your presentation. Thank you very much.
Thank you, Dolores, and thank you, Geraldine, for organizing this and giving me this opportunity. So, hi, everybody. My name is Pilar Marcos Lázaro. Thanks for tuning in. And I'm going to be talking about my PhD, which is the foodborne emergence and epidemiology of Clostridium difficile in Ireland. But first, I'm going to give you a bit of a background on Clostridium difficile because I'm assuming many of you would have never heard of it. So Clostridium difficile is also currently known as Clostridioides difficile. At the moment, it's an anaerobe, which means it doesn't need the oxygen to grow and to survive. And it's a spore-forming gastrointestinal pathogen. In case that some of you are not familiar with this term of spores, I'm just briefly going to explain it. So bacteria can be present in two different forms. They can be present as vegetative cells, which is how they normally would be found. And in this form, they can grow and they can multiply and proliferate. And they can also be present as spores. Spores are the hibernation stages of bacteria. Basically, during this form, they are able to survive really extreme physical and chemical conditions, but they're not able to grow. However, because they're able to survive these extreme conditions, it means that they're easier to be transmitted in this form, in spores. And that's the way that Clostridium difficile is transmitted mostly to humans, is in their spore form. So Clostridium difficile is transmitted through the fecal oral route, which means that the fecal particles pass from one person that is sick with the bacteria to the mouth of another person, which is a new host. So how does this happen? So contaminated feces get in contact with the hands or flies or fields where they grow vegetables, or there's even farm animals, and that eventually gets to the new host mouth, and they get the disease. Even though there's also another way that this could happen, and is that if the previously mentioned hands, the feces, or the flies, or the fields get in contact with the food, and the food gets eaten by the new host. So that's how Clostridium difficile reaches a new host. So Clostridium difficile is able to survive the bile acids in the stomach because in its spore form, as I mentioned, they're able to survive really extreme conditions, and then they reach the colon. So in the colon, they found anaerobic conditions, which means that the spores go back to vegetative cells and they're able to grow and to colonize the colon. And this causes diarrhea and abdominal pain. Depending on the strain of Clostridium difficile, it can produce different toxins, which I will talk a little bit more later. And toxin production can lead to serious intestinal conditions, to sepsis, and in some cases, death. This is what we call the Clostridioides difficile infection. Now, who's likely to get this infection? Well, as you all probably guess, the elderly would be one of the main groups that can get it. And this is mainly because of the changes that they suffer in the gut microbiota and because of their weakened immune system. Then also the immunosuppressed people, or people that have underlying conditions. And lastly, people that are undertaking an antibiotic treatment because there's changes there's a disruption in their microbiota, so therefore they're more likely to get this infection. Clostridium difficile has mostly always been a hospital-acquired infection. It was always thought that it was transmitted only in the healthcare settings, but in the last years, the cases in the community have increased radically. The interesting fact is that the people getting this infection in the community had no contact with anybody from a hospital. So this led many researchers to believe that there was actually a different source of infection, and Clostridium difficile was not just an infection that was transmitted within the hospital. Many researchers started to realize that Clostridium difficile was actually found in food from all around the world. And this led many people to ask themselves, could Clostridium difficile actually be a foodborne pathogen? And if it was, is it a public health concern? So this is where my thesis comes, and this is what I'm trying to answer. The different objectives of my thesis, which would be the first one, to study if Clostridium difficile is actually present in the food chain in Ireland, and to also identify where in the food chain is present. The second objective that I have is, once I've located where the contamination is coming from, are the strains found really toxic, and are they actually relevant to public health? Then it's also important to study how spores can resist different storage temperatures, like freezing and refrigeration, because as I mentioned, that's mostly how Clostridium difficile is going to be transmitted to humans, through their spore form. And I mentioned that they're very resistant to different extreme conditions. So we have to understand how spores behave in a food matrix as well. And we also need to study how optimal temperatures that would be for the growth of Clostridium difficile could affect the spores in a food matrix. So hopefully with all of this in my thesis, I could give a little bit more information about, is Clostridium difficile a foodborne pathogen? So I'm mainly going to be talking about my first chapter, which is the prevalence of Clostridium difficile on farms, in abattoirs and retail foods in Ireland, which is available and you can all read if you want. So what did we do? First of all, we went to different farms and we chose cattle, sheep and poultry, which are the common animals found in Ireland. And we went to 3 different farms per species. And we collected feces, water and soil samples, 10 of each per farm, which makes a total of 270 samples analyzed from farms. Then the next stage in the food chain would be the abattoirs. And we went to 1 abattoir per species, the same species as in the farms. And we collected different samples at different stages of the abattoir. So after the hiding or the feathering in case of the poultry, after visceration, before chilling and after chilling. And in the case of the carcasses, we collected swabs, and in the case of the chicken, we obtained mainly neck skin. And we got 5 samples per stage, which makes a total of 60 samples analyzed from abattoirs. And lastly, the last step of the food chain would be obviously the supermarkets. So we decided to choose 3 to 4 supermarkets per product that we were going to analyze. All these were purchased around Dublin. And we established different categories that we were going to study. So we were going to study meat and meat products, salt leaves and ready-to-eat salads, and dairy products, which had not really been studied for the presence of Clostridium difficile. It was only mainly meat and meat products and salads or vegetables. And we obtained here a total of 240 analyzed samples. So once we had all of these samples collected, what we did first is we did a microbiological analysis, and we were just looking to see, is there Clostridium difficile in the samples? If the answer was no, then that sample would be discarded. But if the answer was yes, then we would also need to provide more information, like, is that strain that we found actually really toxigenic and really relevant for public health? So this is where molecular biology comes, and we did PCR targeting, first of all, a confirmation gene, that is a gene TPI, And this would confirm that what we found by microbiological methods was actually Clostridium difficile. Then we also targeted toxic genes, which would be the TCDA, which produces the toxin A, which is an enterotoxin that mainly affects the intestines. TCDC, which TCDB, which is the one that produces toxin B, and then the genes CDTA and CDTB, which both produce the toxin CDT, which it has been linked to hypervirulent strains. And then we also decided to study the accessory genes because we thought it'll be very interesting. So basically we studied 2 accessory genes, the TCDR, which is a positive regulator of the production of toxins, which means that mutations in this gene could actually lead to a decreased production of toxin A and B. And then we also studied the gene TCDC, which is a negative regulator of the production of toxins, which means mutations could lead to an increased production of toxin A and B, and therefore it'll be linked to hypervirulent strains. So what did we obtain from all of this? Basically from the farms, we obtained a 32% of positives, which is quite high. Mainly we found Clostridium difficile in the soil, followed by the feces, and lastly the water. And from the different characterization that we did, we could see that 41% were actually positive for both toxins, and 58% were only positive for one of the toxins, for toxin B. Now, we would have to look more into this, but this could actually be related to one of the specific strains that Clostridium difficile has that only produces the toxin B. And it has actually been found before to be quite common in Ireland. And then for the toxin CDT, which I mentioned that it was related to hypervirulent strains, 43% and nearly 44% actually were positive for it, while 55% were negative for it, which is also quite a high percentage. Then in the abattoir, we obtained a total of 43% positives. And we did see that as we went on in the abattoir, we obtained less positives. So we obtained most positives in the after the hiding, followed by after evisceration and pre-chilling, where we got the same positives. And lastly, the after chilling, which was the stage where we found the least positives. All of them were positive for toxin B only, and most of them were negative for the binary toxin related to hypervirulent strains. Only one of them was positive. And lastly, in the supermarket samples, we obtained 9 positives, a 3%, which seems like a low percentage, but when you think about it, that's lastly what the new host is gonna eat. The person is gonna access the products in the supermarket. Therefore, 9 samples being positive could actually be considered a quite high percentage. So as you can see, we obtained most of the positives from the salads, followed by dairy products, and lastly, the meat and meat products. They were all positive for toxin B, and one salad was actually positive for the binary toxin, while the rest were negative for this toxin. However, this is quite alerting, because if a person that was in the group of people likely to get this infection were to eat this salad, they would probably get a really hypervirulent strain. And then from our studying of the accessory genes, we saw that 30% had both genes, which means that they didn't have any mutations. And then we obtained really different results for the rest. So just so we all remember again, mutations in the TCDR lead to a decreased production of toxins, and mutations on TCDC lead to an increased production of toxins. So as you can see, we obtain quite a variety of results. So 50% of the samples left were actually negative for both genes. Then 17% were positive for only TCDC, which means there could be a mutation with TCDR. And lastly, 3% were actually negative for TCDC and positive for TCDR, which means there could be a mutation in TCDC. So this is all a theory, and we haven't really gone further into this, but it's something that I would actually like to look more into. So basically, as a conclusion for this chapter, we observe a high presence in farms, in the farm environment, and in abattoirs. We observe a lower presence in retail foods, but as I mentioned, even though the presence is lower, that's the product that ultimately we humans are gonna get and we're gonna eat. So I would still consider that quite dangerous for us. And all isolates were toxigenic, which all might suggest that Clostridium difficile could be a foodborne pathogen. Now, this is only the first and second objective of my thesis. What am I currently working on? So I'm not going to go deep into this, but basically studying the spore viability of Clostridium difficile in different food matrix, especially in meat, dairy, and plant-based, as I studied before. And this is really important because it's going to help us understand if the spores could actually survive through the different freezing or refrigeration obstacles that the bacteria is going to find through the food chain. And what I'm actually also currently doing right now is studying the growth of Clostridium difficile spores in meat, dairy, and plant-based produce. This is basically to help us understand if Clostridium difficile could actually germinate in food if growing conditions were optimal, and then could it actually proliferate and grow. So this is actually what I'm currently doing. I'd like to thank the Food Institutional Research Measure and the Department for Agriculture, Food and the Marine for funding this project. And I'd like to thank the Chagas Walsh Scholarship Scheme as well. And as well, all the symbols that you've seen through my presentation belong to RSAC. As a special thanks to my Chagas supervisor, Declan Bolton, and my UCD supervisor, Paul White, for their guidance and help. And a massive thank you to all the staff and the students from the Food Safety Department in Chagas, which are always such great help. So yeah, I hope you enjoy that. You can find me where you can see in the screen, and I'm open to questions.